Hello biologists! In this screencast, we're going to finish Chapter 3. Let's begin by focusing on the cell membrane. Remember that all cells are surrounded by a cell membrane. The cell membrane is made up of a phospholipid bilayer with embedded proteins that form the boundary of all cells. Phospholipids are molecules with a phosphorus or phosphate component and a lipid component. Remember that lipids are biological molecules that include things like fats, oils, waxes, and steroids. The cell membrane is a bilayer because it has an outer phospholipid layer and an inner phospholipid layer. Cell membranes are also semi-permeable. This means that it will only let certain things pass while keeping everything else on their respective sides of the cell. Every phospholipid molecule has a hydrophilic head and a hydrophobic tail. The hydrophilic head likes water, and the hydrophobic tail hates water. As a result, in an aqueous solution, phospholipids will form a bilayer with the hydrophilic heads on the outside pointing towards the water and the hydrophilic tails on the inside away from the water. The phospholipid bilayer is semi-permeable because it will prevent large molecules like glucose and hydrophilic charged substances like sodium ions from crossing the cell membrane. It will, however, allow small uncharged substances to cross, like oxygen and carbon dioxide. These substances pass through using a process called diffusion. In what we call simple diffusion, substances will move naturally from where they're higher in concentration to where they're lower in concentration. This process requires no energy. In the diagram, you can see molecules moving from where they're higher in concentration to where they're lower in concentration. A real-life example of this happening is heavy perfume or cologne moving across a room, naturally and without energy. Earlier, I mentioned that the cell membrane is embedded with proteins. Some of these proteins are called transport proteins because they help large or hydrophilic molecules cross the cell membrane. They sit in the membrane bilayer and act as channels, carriers, and pumps. They provide a passageway for these substances and can move substances with or against a concentration gradient. Finally, transport proteins target specific molecules like glucose or sodium ions. There's another type of diffusion called facilitated diffusion. In facilitated diffusion, a transport protein is required and a solute will still move from where it's higher in concentration to where it's lower in concentration. Facilitated diffusion also does not require energy. The diagram on this slide shows how facilitated diffusion works. Molecules move from where they're higher in concentration to where they're lower in concentration using a transport protein. Sometimes cells will need to move things from where they're lower in concentration to where they're higher in concentration. This doesn't happen naturally. It requires transport proteins and energy. This process is called active transport, and it's called active because of the energy requirement. Antibiotics can make their way into a bacterial cell by using transport proteins to cross the cell membrane. It goes the other way as well. Bacteria can resist antibiotics by using transport proteins to pump the antibiotics out. This is one form of what we call antibiotic resistance. In the photo, you can see antibiotic resistance on the right side where bacteria are growing all the way up to the antibiotic tablets. Let's turn our attention to eukaryotic cells. Eukaryotic cells are distinct and different from prokaryotic cells in many ways. Because of these differences, eukaryotic cells can resist the effects of antibiotics. Eukaryotic cells have membrane-bound organelles, each of which performs a specific function. This infographic highlights the major organelles of eukaryotic cells. It also shows how we can divide eukaryotic cells into animal and plant cells. We can also add a third category, fungal cells, but those are not shown on this infographic. Let's highlight some of the major organelles found in eukaryotic cells. The nucleus is the control center of the cell. It contains the cell's genetic material in the form of DNA. Reactions for interpreting the genetic instructions in DNA take place here. The nucleus is surrounded by a membrane called the nuclear envelope. Mitochondria are the power plants of the cell. Their job is to extract energy from food molecules. They'll convert the energy in food molecules into a form that can be used by the cell. The singular form of mitochondria is mitochondrion. The endoplasmic reticulum, or ER, are the highways or the pipes of the cell. Proteins and lipids are synthesized here and then directed towards their final destinations. There are two types of endoplasmic reticulum. Rough ER is ER that has ribosomes. This is where proteins are made. Smooth ER does not have ribosomes. This is where hormones and lipids are made. The Golgi apparatus, or Golgi body, is the post office of the cell. Underneath a microscope, they appear as stacked membranous discs. Their job is to package and transport proteins to their final destinations. They'll package proteins in what we call transport vesicles. These transport vesicles are kind of like the UPS or FedEx trucks that take the proteins where they need to go. The nucleus, ER, and Golgi apparatus all work together to produce and transport proteins. The nucleus provides the instructions. The ER provides a site for production. The Golgi apparatus packages and delivers the proteins. The lysosomes are the garbage dumps and recycling centers of the cell. They're full of digestive enzymes that will break down worn-out cell parts and molecules. The byproducts are then recycled or disposed of. The cytoskeleton is the skeleton of the cell. It's composed of protein fibers that perform a number of functions, including cell support, cell movement, and movement of structures within the cell. The cytoskeleton is made up of three different types of proteins: microfilaments, intermediate filaments, and microtubules. In this picture, you can see the protein fibers that make up the cytoskeleton using a technique called confocal laser scanning microscopy. Plant and algae cells have an organelle that animal cells do not have. These are chloroplasts, the greenhouses of the cell. Chloroplasts have two membranes. Inside a chloroplast, you'll find an internal system of stacked membrane discs. Chloroplasts are the sites of photosynthesis. This is where sunlight is captured and converted into a form that can be used by cells. The cells of plants, fungi, and algae can also have a cell wall. The purpose of a cell wall is to provide structure and support. The cell walls of plant cells are made up of a carbohydrate called cellulose. The cell walls of fungi are made up of a material called chitin. Chitin is a polymer of sugar molecules and nitrogen. Biologists have tried to figure out where organelles came from. The leading idea is called the Endosymbiosis theory and states that free-living prokaryotic cells were engulfed by other free-living prokaryotic cells billions of years ago, forming the first eukaryotic cells. We have evidence for this. If you look at mitochondria and chloroplast, they both have two membranes and their own DNA. The diagram on the slide shows how mitochondria and chloroplast might have made their way into a cell according to the endosymbiosis theory.